All right, welcome back for the afternoon session. I have been asked to announce that there are 3 spaces left for the social dinner. So if you did not already sign up and you want to attend, which I highly recommend, it's lots of fun and delicious, then it's first come, first serve. So you probably want to make a mad dash downstairs to the registration desk during the poster session and coffee break after our first talk. So this is going to be a fantastic afternoon. I can't imagine 2 people I would most love to introduce and chair, meaning I can cut them off. So we have Lolly Tyler and Alfonso Caramazza this afternoon. And they're 2 people who need no introduction in the world of concepts, so I don't want to steal any of their time. I'll just turn it right over to Lolly.
There you go. Right. Thank you very much. Thank you very much to the organizers for inviting me. When I got the invitation, I thought, uh-oh, Alfonso and I will go head-to-head again. But it's always very, I'm sure all of you know, that how interesting it is always to argue with Alfonso. He thinks he wins, and you think you win. But it's always very engaging. Anyway, I'm going to talk today about the topic of how we understand what we see. And first of all, I should introduce you to the people who actually did some of the hard slog. This all started with Helen Moss and I talking about category-specific deficits about 20 years ago. She left to become an author, a very successful children's writer. And then I went on to work with Kirsten Taylor, Alex Clark, Barry Devereaux, Billy Randall, and Paul Wright, and you'll see their names popping up as we go through the talk. Okay, so I think we could say that we could be forgiven for thinking that one has to assert that objects are more than just visual images, that they actually have meaning. And when I talk about objects, I'm talking about everyday objects. I'm not talking about faces. I'm not talking about places or anything. So this is a very restricted domain that I'm talking about. But anyway, as I said, that the objects are visual images that have meaning. And I'm going to concentrate today on trying to understand something about the relationship between vision and the meaning of objects. So how does the brain transform low-level perceptual inputs so that they become, yes? Oh, I thought already. Too soon, too soon. How does the brain transform low-level perceptual inputs into meaningful object representations? So how does perception become conception? Because after all, that is the goal of the system, it would seem to me. But this issue is largely ignored, and the focus of research has mainly been on vision or semantics, but not the 2 together. And in fact, the main bulk of the research has been on vision, on the visual properties of objects, with some work on the semantics of objects, but it's very often, not always, but very often sort of incidental to the major question of how vision leads or delivers to us meaning. It's usually in the context of wider issues about semantic organization. Anyway, our goal was to try and understand this relationship, and to do that, we decided that we had to integrate a model of vision and a model of semantics. And this is the model of vision we chose, plus the model of semantics. So our model of vision then was based on primate neurobiological models of the ventral visual stream. So the model that is mostly taken for granted, which is that there's a hierarchical system with increasingly complex visual representations going from posterior sites along to anterior sites in the ventral stream. I'll say more about that in a minute. Anyway, and then we put this together with our model of semantics, and there's lots of reasons why we did this, but basically, we needed to know what we meant by semantics. And to do that, we had to define it for all the weaknesses that are associated with defining semantics, the limitations, I should say, but we defined it in terms of a computationally explicit feature-based distributed semantic system, which looks something like this. And again, I'll talk a little bit more in detail about that in a minute. And then we tried to develop a dynamic neurobiological account where we integrated the visual model with the semantic model, and we used fMRI and MEG to determine how visual inputs are transformed into meaningful representations in real time. Okay, so for those of you, I'm sure everybody knows all this, but for those of you who don't, the idea is that what happens in the visual system, so we're talking about visual object processing now, what happens in the visual system is that simple features are represented in more posterior regions with increasingly complex feature conjunctions as you go more anteriorly along the stream, culminating in the perirhinal cortex where the most complex feature integrations occur. So this delivers to you the notion of over time and over space, going from posterior to anterior regions, coarse to fine-grained representations, and we'll hear more about this in a minute. Another important aspect of visual object processing is this coarse to fine-grained visual representations is achieved through recurrent activity. So when you see a visual object, you have, in the first 50 milliseconds, you have activation, or the engagement of occipital cortex. Activity runs forward within the first 100 millisecond sweep, and you get recurrence where activity goes back, and you are getting this feed forward and feedback system. And the idea is that the fine-grained representations require more of this recurrence than do coarse-grained representations. Okay, so the question we asked then was, does conceptual processing, so the meaning of an object, the construction of a meaningful representation, and construction is used advisedly. I don't think these representations sit anywhere, and I'm glad that some people are not in the audience when I say that. So the question then is whether conceptual processing shares similar principles. So as the visual system, so does conceptual processing also show this coarse to fine-grained representational development throughout the ventral stream? And does this evolve through recurrent processing? So if we look here at posterior sites going forward to anterior sites, in posterior temporal cortex then, we expect these simple combinations of conceptual features just as in the visual system. And also there are these simple features enable fewer integration demands. So this is sufficient for coarse-grained and category-level representation. In the anterior temporal cortex, particularly we're talking about the perirhinal here, you get more complex combinations of conceptual features if the conceptual system has the same principles as the visual system. This is what we'd anticipate. So it can cope with, it has the capacity for greater integration demands, more complex, fine-grained representations can be developed in this site. And these are necessary for object individuation. Now, a lot of what I'm going to talk about today is about the difference between categories and objects. And it would seem fair to say that, in fact, most of the literature on object processing does focus on categories and not on individual objects. And the representation and the construction of the representations necessary for category structure and for object individuation, for determining the representation of a unique object, they're complicated, and we need to look into that. Okay, so the first experiment we did a long time ago in 2004 to look at this, to look at this coarse-grained versus fine-grained representation was an fMRI object naming study. Now, the issue of tasks has been talked about a lot in the morning sessions. And I agree with that. I agree with the importance of thinking about the task completely. So what we've done, and all the experiments I'll talk to you today are like this. We've used almost always, apart from this first two experiments, almost always we use object naming. And the reason for using object naming is because we think that you need to tap. If you need to tap into certain kinds of representations, this is a task that you need to use. So you can't tap into category structure or similarity structure using some of these tasks. Sorry, you can tap into similarity and category-based structure using a variety of tasks. But to look at individual objects and how they differ from their category members, you need to use something like object naming, individual object naming. And so that's why we use this task. In this experiment, however, people named the same pictures in two different tasks. So first of all, they were asked to name the object as a living or non-living thing. And we argued that this was going to engage coarse-grained representations. You only need to know something about the similarity structure of each domain to be able to make this decision. You don't need to know a lot of fine-grained detail about the objects. And we compared activation when people were doing this task with when they were doing basic level naming on the same objects. So it's a fairly simple experiment. You see a cross, you see an object, etc., and you name it. And as I said, this is fMRI, this first one. And what we found in this experiment was, if you look here at domain level naming, you can see that activation in both hemispheres is fairly posterior in comparison to basic level naming, where you see activation going from posterior to anterior sites, exactly the same stimuli. And you can see we've tried to plot the effects along this posterior to anterior axis along here. And of course, oh yes, okay. So you can see that domain level naming is the red one, and that the activation for domain level naming goes down as you go more anteriorly, but the activation for basic level naming stays high throughout the ventral stream. So we argued then that these effects couldn't be purely visual because it was the same objects in both tasks, and that similar to perceptual processing, different aspects of conceptual representation are constructed in different ventral stream regions. So in these posterior sites, you get these coarse-grained representations giving you category information, and in these more anterior sites, in this case, we didn't see the perirhinal particularly, but in these more anterior sites, you get more fine-grained differentiation enabling you to represent individual objects and differentiate them from their neighbors. Okay, the next question we asked was, are these effects underpinned by recurrent activity, just as this coarse to fine-grained representation is in the visual system? So then we carried out, Alex Clark carried out a MEG version of the same fMRI study using the same stimuli, and again doing basic and domain level naming. Okay, so when he compared then the MEG data for basic and domain level naming, he found that there was an increase in global field power in MEG sensor space within this time window, 170 to 258 milliseconds from the onset of the object. This shows you the effects in sensor space, and this was the source localization. Okay, the source localization I've showed you here in order to show that you get fusiform areas and anterior medial areas, and also that we see some frontal activity. Now, what this graph here shows is effects in each of the three regions that we took as ROIs. So we did this analysis using a priori ROIs taken from the activated regions in the fMRI study, because we wanted to see what was going on in those specific regions. So you see occipital cortex here, fusiform here, and anteromedial there. And so that's, this occipital's black, the blue is the fusiform, and this is the orange is the anteromedial, and the green is the frontal cortex. This ROI comes from one of Barr's studies. Okay, so this is the difference then, remember, between basic and domain level naming. This is greater effects for basic compared to domain. And you see that, that this is the window where we saw these effects, this basic versus domain increase. And you see that for the anteromedial region and the frontal region, you get a surge in source strength within this window, followed by an increase in the fusiform, suggesting recurrence. You only see this in the left, not the right, probably because this was a naming task. So we wanted to go further with this and determine whether or not the relationship, this recurrent activity that we claim that has been observed in visual processing was also evidence in this task for basic level naming for fine-grained differentiation. And so we looked at whether or not any of these ROIs, we looked at each pair of ROIs, we looked to see whether there was significant synchronization, phase locking, during basic level naming. And we found that it was only between the fusiform and this anterior site that we got significant effects of phase locking, indicating increased recurrent interactions in this frequency band here. And again, within the time window where we saw the significant effects of basic level naming. Okay, so we see then in these two studies a close correspondence between the processing characteristics, if you like, between perceptual and conceptual processing in the ventral stream. Now, to take this further, we decided that we couldn't really do anything unless we knew what we were talking about when we were talking about semantics. So, to that end, we decided to look to feature-based models of semantics. Alfonso was an early adherent of this kind of approach, I want to point out carefully, because he isn't so much anymore. Okay, so there were various feature-based data sets around, but we use this. We use McRae's. His is the one that's most highly developed, but we also constructed our own. Anyway, the whole point here is that you get semantic features, which are obtained from large-scale norming studies, and this is 500 or 600, 700, 800 concepts. And so you ask loads of people to write down all of the features that they associate with this concept. And then you develop a matrix where for each concept, you can determine whether or not that concept has that feature or not. And so you get a vector that corresponds to the feature space of that particular, for every particular word. And it's this kind of model that I'm going to talk about and use in the experiments I'm going to talk about. Okay, but just having a feature matrix and just having a vector of an individual object isn't enough. So what we've done is we've developed a lot of different measures of the relationship of the kinds of features that we've got and the relationships between features. So we think that looking at the relationships between features is really important, because a concept's meaning is not just a list of features. It's also about the relationship between features within the concept and the relationship of features between concepts. So you can see here that this gives you some idea about how we can visualize these sorts of models. So object meaning is represented as patterns of activation over many nodes like this, many more nodes, in a distributed network. And the nodes or groups of nodes correspond to semantic features, objects, categories, depending on how you cluster them. And this is a computationally explicit model, and that was what we wanted. So you can see here that for the individual nodes like this, right, they can vary on the scale of distinctive, that is, they only occur in one or two concepts, to shared, where they co-occur in many. But we also look at the relationship between features, looking at the correlational strength between every pair of features. And the thickness of the line indicates how strongly two features are correlated. So that's another variable that we include in our matrix of measures. We also include a whole load of other things, but these are the key ones that I'm going to talk about or use when I talk about the experiments today. So we obtain then, as I said, quantitative measures from property norms for objects and categories. And I have to say that we've got extensive behavioral evidence validating this model, that these kinds of measures derived from property norm data are important in interpreting behavioral data across a wide variety of different kinds of data sets. And what I'm going to do now is use this model to explain the basis of category representations and the basis of individual object representations and how they're differentiated from their category neighbors. So I suppose what underpins all of this research is the interest in, there have been wonderful developments, I think, particularly starting with Jim Haxby's work in 2001, that have enabled us to really look at different aspects of object representations. But one of the things that's always left me feeling dissatisfied is that I never quite know what people are claiming about when they claim that similarity underpins X or Y. Because very often you've got no idea what the basis of the similarity is. You know that things are clustering together in semantic space or whatever, but you don't know why they're clustering together. And somehow or another, the fact that you can attribute a label to that cluster means that you've explained it. And I never feel that it explains it to me. So that's why we've gone to all this trouble. And it is a huge amount of trouble. Okay, so that's the reasoning behind all of this. And now I'll tell you some of the results. So the first thing we wanted to know was, well, in fact, you've got all these quantitative measures from this feature-based model. Do these feature statistics actually explain effects in the ventral stream? Okay, so we did an fMRI study, basic level object naming, and we used feature statistics from the property norms and other variables for a large varied set of 412 objects. So the great thing about this is every object has got lots and lots of properties, and you can develop models based on each one of these properties and determine where, if anywhere, you see effects along the ventral stream. So what we were hypothesizing then was if activity along the stream reflects our feature statistics, we should see feature sharedness, which gives you category information, effects in the fusiform. This should overlap with known category effects for animals and tools, for example. And you should see within category differentiation, so object-specific properties. Now we're looking at individual objects, and these effects should be in the perirhinal cortex. Okay, so we got the fMRI data for all of these objects, and we did a PCA analysis and looked at the effects where these principal components were correlating with brain activation. So there are two main components I'm going to talk to you about. One is the sharedness component, and I hope you can see the colors here, because basically what I'm showing you is in blue that's these, right? You can see this is where you have maximal effects of shared features, okay? And if you look below, which is where Alex Martin, one of his review papers where he showed the effects of animals and tools in the fusiform, you can see that the blue, the sharedness, is similar. So this is lots of sharedness, and that correlates, that overlaps with the animal ROIs that he finds in the fusiform, in the lateral fusiform. Hang on a minute. Sorry, I apologize. I've completely got that the wrong way around. The blue is medial, and that's correlating with the, hang on a minute. I'm all right, aren't I? Yes, sorry. I'm looking at things sort of sideways here. Anyway, the fusiform is outlined in another color there, which, unless I'm going to go in colorblind, I can't really see. Okay, the point is that sharedness, the principal component of sharedness, shows an effect which overlaps with tools in the fusiform as seen many times in other studies, and where you get many shared features, which is true of animals. Have I just said this twice? Yeah, I apologize. Right, I think I'm going a bit wonky. I would love some water if there was some. Anyway, I'll carry on though, and try and get it right in the end. Okay, everybody knows what I'm talking about. I'm moving on because I don't like that slide anymore. Okay, right. Now this gets even more tricky, okay? I better get over here, and then I'll fall down this thing here. Oh, thank you very much, Alfonso. I'm not usually such a twit. Thank you. Okay. What we're doing here is we've got this, we're looking at activation values along the lines transecting the fusiform gyri from the medial to its lateral extent. And here's the effects. Here's the effects that I'm going to explain to you. All right, first of all, you only look at the purple and the green line. Let's look at the green line to start with. This is the sharedness component. So we're mapping this from the lateral to the medial extent of the fusiform. And you can see that you have a peak of sharedness in the lateral extent. And the weakest effect, so that's less shared, right, is having an effect in the medial extent here. Okay, now overlapping with this, you see living, non-living. So we also categorize these objects as living and non-living, and you can see that they conform very closely to what we find for sharedness, which is what you'd expect. So now you might say, well, why bother with all of this feature-based stuff when we've got category representations and we understand them in the first place? And it's because we can explain something else with the same model. So parsimony and all that kind of stuff. Occam's razor comes to mind. Okay, so now we're going to map the object-specific component, okay, from the fMRI study. And the effect of this ease of this is a particular way of looking at the feature-based norms, which gives you a numerical value for the ease of differentiating between highly similar objects, semantically similar objects, right? And you can see that this is located in the perirhinal cortex. So the differentiation then between semantically similar objects that you need in order to distinguish one exemplar from its category members requires activity in the perirhinal cortex. So the point here is, with a single set of principles, we account for different aspects of meaning. And in this field, that seems to me to be something, because it doesn't happen all that often. Okay. So in another study then, so we've, all of that I'm going to talk about is about this object and category effects, and can we understand them, and what do they mean? So another study used RSA, Nico Kriegerskort's very illuminating methodology to relate the cognitive models to patterns of neural activity. So this, again, used a large, diverse set of objects with many different properties. And this is the wonderful thing about objects, that using the same objects, you can look at them in lots of different ways and see where different properties have an effect in the brain, or correlate with activity in the brain, I should say. All right, so we generated many different models, visual, categorical, animacy, et cetera, and we used a whole brain searchlight so that we could test each model against the brain data at every voxel on a brain-wide basis. So this is Alex Clark, who has done a lot of this stuff. Okay, so we have models like this, which are visual models. We've got semantic feature models based on the semantic feature data. And you can see that in these vectors, the semantic vectors for each concept give you both category structure and the information that you need to individuate between members of a category. And then we tested lots and lots of different theoretical accounts of the differences that you find in the fusiform, right? Differences between objects. So like the animacy, where the biological model so-called here. So this is living, I think this is living, non-living, and plant life. And anyway, I can tell you about the details of that afterwards. But it certainly includes living things, plant life, and artifacts. And here we have separable categories, and here we have just an object category RDM. And what these RDMs are modeling then is, for example, with this, it's saying, I'm only interested in regions of the brain for which animals are clustered together and everything else is irrelevant, okay? Whereas this is categorizing these three different category types, right? So it's looking for regions which differentiate between them, and similarly for that. Okay, so you test these models against the brain data, and this is to give you a little bit more insight into what this feature-based model is. So you can calculate, or Alex can calculate, the similarity or the overlap of semantic features between all pairs of objects. To get this, and as I said before, this model captures both categorical similarity between objects, so objects from from similar categories of similar features, and within category object individuation. Okay, so what do we get when we test this brain-wide, voxel by voxel, brain-wide across the brain? So the EVC model, so the low-level visual model, gives you effects in the visual cortex, and if it didn't, we'd be in deep trouble. And then semantic features, the semantic feature model gives you effects in the anterior medial temporal cortex. And this is I should have pointed out that these are the unique effects of each of the models after removing the effects from all the other models. So these are unique effects. Okay, and then here, category effects in the fusiform. Again, this is the animal, plant, biological model, again in the fusiform, and similarly for animals. So what we can say as a result of this study then is that you see in the fusiform a largely distributed categorical organization, but that the organization, the regions can overlap as a function of different kind of modeling that you test against, so that you have these distributed categorical representations. But in the anteromedial temporal cortex, you have only these regions are sensitive to object-specific effects. Now I'm going to talk a little bit about neuropsychological data to try to convince you of the validity of this kind of account and why it's worth doing. Okay, so for many of us, there's a phenomenon of category-specific deficits associated with damage to the anteromedial temporal cortex that has been a puzzle for a long time. And we could think that we can actually understand why this has occurred. Okay, so first of all, this is a patient, and this is their drawings from memory. Now, these are non-living things, and I expect that you can actually figure out what they are. And I'll give you the names, and you can determine whether or not you should see your local neurologist. Okay, right. Now we're going to go to living things. Any candidates? Okay, so I think everybody would agree that it's jolly difficult to figure out what each of these objects were, although you know that they were living things, and that you knew they were living things because they had shared properties were remembered by this patient, whereas the distinctive properties which differentiated between highly similar objects were not remembered. And this is associated with damage to the to the anterior medial temporal cortex in this patient. And of course, that isn't very illuminating, really. Anterior medial, a huge area, blah, 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 blah. So we tried to sort of refine this by taking patients who had damage to the anteromedial temporal cortex, dividing up the subdivisions anatomically. And what we found was, sorry, I'm going to do this. We found that increasing damage, increasing perirhinal damage, so we managed to isolate the perirhinal and the entorhinal and this, that, and the other, and it was only increasing damage to the perirhinal that actually was associated with problems in naming objects that were semantically more confusable as determined by our feature norms. And I need to go back to explain why this is the case. And it's the case, so from the property norms, what we know is that animals have got many shared properties. They're easy to categorize, but they have few distinctive properties. This is in the norms, hundreds of people, hundreds of concepts. So within category differentiation is difficult because it's the distinctive information that enables you to differentiate within a category. Whereas non-living things have few shared properties, they're harder to categorize, but they have many highly correlated distinctive properties. And this enables within category differentiation. And so when you have damaged the perirhinal, and you can't do this within category differentiation, that's why it's the distinctive, you get the category information out, and the patient can remember that, but you can't do this fine-grained differentiation that you need to differentiate objects, similar objects from each other. Okay, right, what time is it? You see, I forgot to put my timer on.
About 6 minutes.
Oops. Okay, now, so an important aspect of all of this is when does perception become conception? So when in the real-time analysis of the input do you see these visual and semantic effects? So we're going to use MEG then to look at the real-time spatiotemporal integration of perceptual analysis and semantic knowledge. Again, an experiment that Alex did. He had the magic touch when it came to MEG data. So we know that object recognition involves the rapid transition from coarse to fine-grained representations. But what we don't know is whether these effects over time are better modeled by visual or semantic models of objects. So again, we did a MEG study with lots and lots of objects, and people named the objects. And in this experiment, we combined a biologically inspired visual model, HMAX, and our feature-based semantic model. And we estimated the fit of each model at each millisecond over time, and asked, when does each of the models best fit the MEG data? So this is what you see. So this is time here. And these are the different models we tested. So when you're looking at the low levels of HMAX, so HMAX, for those of you who don't know, is a hierarchically structured neurobiological model of the visual processing in the ventral stream. And this is one of the lowest, the earliest perceptual levels in the hierarchy. And you see that most of these models have a maximal effect between 50 and 150 milliseconds after object onset. So you see this effect around 120 milliseconds or something like that. And now, so this is only the very lowest level. Now we're looking at the whole of this model with all its different levels, which approximate the complexity of the visual representations along the stream. Right? And you can see that it's doing better with high-level representations included. And then this is when you've got HMAC, so the visual model, and also the semantic model. And you can see that you do better again, the tip is higher here, and also, unlike this model, for example, you've also got significant effects occurring later on. So you can see that more clearly here. So this is when you've got the full HMAC, so you're capturing the complexity of the visual representation along the stream compared to very early visual effects. You can see that you better model the MEG data in this early time frame, 120 milliseconds. And then this is showing you the sort of recurrence, if you like, of semantic information occurring after this initial sweep. Okay, now for my sins, the last thing. Okay, what we've been doing, this is actually Barry Devereux's work. What we've been doing, so the interesting thing is that, we're talking about vision to semantics, but it leaves open the question of how this is achieved. How do visual inputs become semantic features, for example? So how do visual features become semantic features? So we've thought about this quite a long time and never had any ideas, really, until these deep neural networks came along, which we've been exploiting to be able to try and understand the relationship between visual representations and semantic representations. So what Barry has done then is he combined a visual model and a semantic neural network model. So the visual model, this is the DeepNet visual model, and its task is to accurately label objects. And there's a question that it doesn't actually capture meaning from vision. What it's doing is providing a unique label, which looks like that's semantics, but in fact, you don't find with these models that they're clustering, the labels don't cluster semantically, and so they need additional training. This is work that Nico's done with some other people. They need additional training in order to be able to capture the sort of basic properties of a semantic system. Okay, so this is the deep neural network. This is pre-trained. So this network is pre-trained. And we use the pre-trained network on our set of 600 objects or whatever it was. And we got values for each object at every level of this system, right? Now, what we did then, oh, hang on a minute. That's not the right place for that. Okay. What we did then was to add this model, right, which is a recurrent neural network model based on the property norm data, okay? And we took away the labels, or Barry took away the object labels here at the end of the DNN and attached this to it. So you've got this model that's learning the visual and semantic mapping as a function of the representations that have been derived from all of this processing at around here. Okay, so it's high-level visual input. So just to give you an idea about what's happening in the attractor network and the semantic model, so this, sorry, I haven't done this right. Right. This is learning over 20 time ticks. So it's learning over time to associate features, semantic features, with these visual representations, these visual features. wrong way around. Okay, so now we're looking at the semantic network and how it's accruing information over time, how it's learning, if you like, this mapping. And what you find is that over time here, that you start off with sharedness. So this is mean feature activation. So features that are highly shared are those that are coming online in the attractor network the earliest. And then we see later effects of all the other networks, of all the other features. Right, that's the most important point to take away from that. And here we see an example of the semantic features which are most strongly activated by object images. So you can see then that these are the activating images, and these are the semantic features that are activated by these and many other images. So these are features that are shared then, many of which are visual, but they're shared between many objects. Okay, so these are the right. So these are the general properties of this attractor network with its inputs from the visual model and the kinds of representations that are coming online. So after the attractor network had been trained then with this visual input, what Barry did was, so this is the DNN, the different levels from low-level vision up to higher-level visual representations of the DNN, and then these are the time ticks of the Semantic attractor network. And for each of these, Barry constructed an RDM, right, from the model, and then correlated those RDMs with MEG activity, and sorry, not MEG, fMRI activity. And he did that in particular regions of interest that were important for developing this kind of model, and also whole brain searchlight analysis, which I won't talk about. So this is looking at the back of the brain then at areas 17 and 18. And what you can see here is this is the correlation, these are the correlation values of how well the model that's constructed for each, the RDM that's constructed for each of these levels in the system correlates with brain activity. And you can see that it's this early visual processing, right, from the DNN that's correlating best with activity in these areas. Now we're further along in the fusiform, and you can see now we're beginning to see the early stages of the semantic model having an effect. And remember that the semantic model, these are shared features that are coming online earliest. And then when we go to the perirhinal, we can see that you're getting later effects of the semantic model, and this is where you get fine-grained differentiations, where you get distinctive features, et cetera, et cetera. Okay, so I'm going to finish now. So the layers of the visual DNN then only account for activity in visual regions. And this posterior ventral temporal activation is best explained by the early stages of the semantic network, where you get shared general superordinate category information. But full activation of detailed semantics of specific target concepts best accounts for activation in bilateral perirhinal. So what we find in this model then is when you've got explicit models of visual and semantic representations, you can account for the fMRI pattern information throughout the ventral object processing stream. And my final slide is, okay, so object meaning, which I think should not be ignored in our trying to understand how objects are processed in the brain, are processed within a conceptual hierarchical recurrent system in the ventral stream, similar to visual object processing. And different types of object meaning are constructed through the computational properties of different regions within the ventral stream. And I think very importantly, that this enables flexibility of semantic representations. We don't talk much about this, but I think that one of the hallmarks of semantics is that we can use concepts to mean different things depending on the context in which they occur. And that has to be enabled by a brain that's flexible enough to do that, but also knows its limitations. So I would argue then that the semantic feature model that I've told you about gives a principled approach to understanding how the brain represents different kinds of semantic information. Thank you. Right, shall I put my helmet on?
Hi, thank you for the great talk. So I really like your general approach of trying to understand concepts in terms of elementary features that can be combined in different ways. So you have sort of a generative model for creating novel concepts. One thing that is not clear to me is how do these the semantic features in your model, it seems to me like the features that describe the concepts are themselves complex concepts.
Oh, I should have said this. You're absolutely right. They are, of course. And I should have mentioned this health warning right at the beginning. So it's not that I believe that there are concepts, that there are features instantiated in the brain, but that they're proxies for much more complex representations, which we can't get access. I mean, so far we haven't been able to get access to. So this is the sort of information that we can get from people. There's probably other ways of trying to drill down and understand the nature of these feature representations, but we haven't been able to think of it, and I don't think anybody else has really. But absolutely, yes, thank you for bringing that up, because I do not believe that there are features like this. I mean, there might be at some level of representation, but they're not the primitives.
So do you think there is a potential for trying to redescribe those features in terms of more elementary perceptual motor features, multimodal representations that are, say, because there seems to be a gap, like when you're trying to explain how semantics emerges from vision, right? But there seems to be a gap in between the perceptual representation, the perceptual feature, and then going to the semantic feature, which,
yeah.
So I'm trying to think, how do you bridge the perceptual level of neuronal representation and the semantic level of feature representation? Is it a matter of combining multimodal perceptual information in different patterns? So do you have any ideas in terms of how to bridge that?
Well, in the DeepNet study that we did, what happened in the attractor network was that it was initially driven and dependent on these visual features, right? Shared visual features was what was sort of driving the initial stages, right? But through recurrence and backpropagation, right, you get other features that those shared features are correlating with, right, in the network. So they're all coming online too. Now they're coming online via the visual system, but they're not visual. Now, with respect to multimodal, I mean, all of the stuff that we've done has been about naming objects, static objects. Now, if we were doing something different, undoubtedly, I think you would get different kinds of other regions coming online. Probably multimodal, but certainly sensory motor regions of different types. I think that all of that kind of approach can be accommodated within this kind of network modeling.
So I wanted to ask about your distinction between coarse and fine-grained. So you're making that distinction on the basis of the task. Okay, so if I can make
sorry. I'll wait. Sorry.
So if I can make a fine-grained distinction about something that requires me to know that this is a panda, as distinct from a coarse-grained that this is an animal. Okay. what I'm trying to do is reconcile that with ideas that I've seen elsewhere that talk about representation as you go through the system, going from, in effect, if you think in terms of receptive fields, smaller, what they call fine-grained spatiotemporal fields, to, as you progress up the system, coarser-grained. And so there's fine and coarse grains seem to be being used in opposite ways. And some of the distinctions, I mean, if you think about high sharedness coming online soonest, I don't need to know that this is a panda in order to recognize that shared feature. I can just look at a bit of fur, for example, and see that that's an animal. So I just wanted to know whether I just want to know how you would reconcile what you're referring to as fine-grained and coarse-grained representation with this other approach, which says that, at the retina you may have, these smaller receptive fields, and then as you progress up, you're getting ever-increasing receptive fields. It seems to me that representationally that's the opposite to what you're saying.
Well, it is, except that there are lots of theories of visual processing. And this hierarchical model, which says that, which claims, and there's lots of evidence for it, that you have more coarse-grained features originally, or in the early processing of the visual input, right? Because of the neurons and what they can do in those posterior sites. Whereas you get fine-grained information, more fine-grained, because of the neurons being more complicated, being able to integrate in different ways and more frequently or whatever, in more anterior sites, that that gives you the basis for combining lots of different or more different information. I mean, the thing about the visual system, seems to me, is that you can cut it up in lots of different. I mean, there are very many ways of explaining and describing what's going on in different regions. What we're trying to do is to explain or use models that give you this sweep of information, because you've got to reconcile the effects that you get posteriorly and the effects that you get anteriorly. So, they exist. We see that all the time. And how do you put that together? Well, you can put it together with a kind of hierarchical account. So it's, yeah.
Hi, Lally. So, one, I wanted to congratulate you. Of all the people who talk about anterior temporal lobe, I think you're the only one who actually tells us where you're talking about, which is a big improvement. So let's talk about perirhinal.
Okay.
And we've discussed this previously, but it seems to me that one of the difficulties with this kind of it's not the difficulty with a hierarchical model. It's the difficulty maybe where that takes place and what actually is happening all the way downstream. So one of the things I've asked you about before, the amnesic patients, bilateral medial temporal damage, including perirhinal cortex, and they can name objects. Same thing with temporal lobectomy patients. So there's
specific objects. So they can do this, they can
basic level naming is...
Basic level naming of animals too.
Sure.
Well, that is different though from the, the patients who, the well-known also category-specific deficits for animals in that region. In terms of damage, involved in damage to that region.
Right. So actually that's not what I originally wanted to ask you about, but so is the model that it's only category specific, as opposed to, I thought all the features were being integrated there, independent of category?
Yes, they are all being integrated there, but there are consequences depending on the category to which those features belong. So when you need, with tools, for example, you don't have a lot of shared properties. So they're not going to be indistinguishable and require this additional processing effort, if you like, to differentiate them.
So I can do basic level naming of tools without
your perirhinal, yes. That's my prediction anyway.
So let me just give you one alternative explanation for the data on basic object naming versus the category distinction. Given that perirhinal cortex is kind of the gateway to the hippocampus, this information has to reach hippocampus if you're able to remember, which people can. Then those two tasks could be seen as, you know, classic levels of processing manipulation. Naming items at the basic level leads to better memory than a task that kind of interferes with basic level naming, which is what I think happens when you force people to do this more unnatural category naming. Either way, it's still kind of a levels of processing argument, and therefore you're getting the activation all the way downstream because that's better encoded because you named at the basic level.
Yeah, so you're interpreting this in terms of the task, what you need to do, operate on the stimulus. And what I'm saying is it depends on the nature of the representation, right? So that, you know, there is this general difference between living and non-living things. And what I'm arguing in terms of this sharedness and distinctiveness and the relationship between them, which makes inherently living things more difficult to differentiate from each other. Therefore, and not tools don't have that problem. Therefore, if you give somebody a category judgment, they're going to be fine on both. If you ask someone to do basic level naming, that's where you'll see the difference, with damage in the perirhinal affecting the animals and not the tools. Now, you could say that this is a task effect, but then I think you have to explain why, right? Why you get this difference for animals and tools. What's the basis of the difficulty? What's the basis of the difference? You know, you know, you're probably right, but I'm just saying I don't know what the basis of the difference is that's going to give you this differential effect.
Just one more quick question. So in terms of your thinking about semantic system, is it all temporal lobe?
Well, you see, no, it can't be, as you and many others have shown. But I must say, when we do these experiments, I'm showing you all the data that there is. Now, there's one thing I'm brave to talk about, right? But so in all the experiments we've done, we always do a whole brain analysis, even if we do ROIs. We never find anything other than the ventral stream, except in this deep net analysis. So, and I don't know what it means yet. So when Barry has, so I showed you the ROIs, but he's also done whole brain searchlight. And in the whole brain searchlight, you see, the first time we've ever seen it, I know everybody else sees it, but you see this swathe along the dorsal stream, as well as the ventral stream. So there's obviously more to, well, obviously you all know that there's more to this than I'm talking about. But you know, I've never had to face it in my data, really. And now we have to think about this and try and understand, you know, it was basic level naming again. So maybe it suggests to me that there's something about the modeling we're doing, the models, that we're missing. There's something going on here. So, yeah.
So I just had a question about, a theoretical question about the interface between vision and semantics. And the way in which you framed it was that it was feed-forward.
Well, it can't be, can it?
Well, okay, so I wanted you to elaborate on this because, you know, one way to think about it is that the visual information is not semantically interpreted and you get a high-level visual representation and then you figure out what concept corresponds to that.
Yep.
Alternatively, it could be that the visual information is already semantically interpreted, and the task of the system is not to figure out which concept corresponds to the high-level visual input, but to figure out, essentially to access the vision from the semantics, given some initial guesses that the system already has. So have you kind of thought about the directionality of how this might be working in the context of that model that you proposed?
Well, in everything apart from, so it's the deep net that's feed-forward, right? But all the other stuff we've done, we're assuming an interactive, you know, feed-forward, feedback, a recurrent system, because that's what the neurobiology of the visual system looks like. So, and we see, you know, that So, and we can see that in the timing, right, in the MEG data as well, that you get certain things coming online in the first sweep, and other things, other kinds of information coming online when you're after you've got recurrence. So that seems to me to be much more plausible with respect to unifying visual and semantic models. But the deep net, these deep net visual models are feed forward, and why we did this last experiment was to see how far we could go into, because there is this tantalizing issue about if you believe that there's at some level of the system, there are visual features and there are semantic features. There's certainly something semantic, even if you don't have a model of what it is. They've got to connect. And where does that come from? How does the brain do that? So that's what the attraction of doing this DeepNet plus visual attractor network was for us. But I agree with you. I mean, one is already skeptical about the visual model, given that it's feed forward. So I agree with you.
Hello. I've asked this question other times, not to you, but in this meeting, the question of recurrence, and this is a concept I have a hard time understanding. So it's a two-part question. First, just a clarification: when you show those data, this activations, whatever they are, going up, peaking, and then coming down, and the other one coming later, you take later to mean recurrence. And I'm curious to know what the logic for that interpretation is, whether in fact there is evidence that it's recurrence and it's not just late activation. And then supposing that in fact there were some way to link these things, I have a hard time with recurrence. And it was tied deeply with the claims you're making about concepts, perception, and so on. On the one hand, you say your model is layered, so that in fact different levels have different kinds of representations. Second, if that's wrong, then I stop my question. My assumption was, in fact, that you were saying that you go through these layers and you're essentially getting different, more complex, whatever kinds of stuff, and that the connections from one layer to the other is possible because the information at level 1 is commensurate with level 2. In other words, whatever it is I compute at level 1 is of a sort that allows me then to map onto whatever I represent at level 2. And then from level 2 to level 3, the same thing. Now, on this logic, the information at level 1 is not commensurate with level 3 unless I already have a linkage between 1 and 3. That is, I already have some way of parsing the information such that it can map onto this more complex representation upstream. If this reasoning is at all plausible, then recurrence is not possible. That is, recurrence as information-based recurrence is not possible because as I go up there and I say, oh, I have a dog. Well, okay, so what the hell do I look down at when I look down in the visual area? What am I going to feed back onto for many reasons? One, because of the fact that visual field, I mean, the information you're picking is of different sizes, and it's not clear to me how that model makes sense.
Okay. Right. This is an interesting question, I agree. I think logically one would need to posit is that there is information carried forward that is consistent with all levels. Right? Now, this doesn't have to mean that it's veridical. It's exactly the same as in level 1 or whatever. It just has to mean that it's mappable. And I mean, you do see, I mean, if you don't like recurrence, the thing is that you do, there is just evidence here. You do see increased activation in posterior sites after you've gone all the way forward. Well, there's got to be doing something. And because it's there and it's not somewhere else, right?
Right. I have no problem with the first answer, but that changes your model completely. That is, now you're going to have to come up with some articulation of what you call features, properties, whatever, such that they allow you to do the mapping across levels. And then with respect to the second one, I also agree. I mean, it's later, but it doesn't mean it's recurrence. So here's a possible thought. So I start activating at some visual level, something comes in, I start processing it, and then information at some point in time is sufficient to go forward to the next level. And it's forward and so on until, probabilistically, statistically, I reach dog. It doesn't mean that I've seen everything in the picture of dog. That is, in other words, I could continue processing the information at the lower level and show this greater peak later. So what I'm trying to say, it's a very stupid thing I'm saying. What I'm saying is that time doesn't mean recurrence.
Yeah, no, you're absolutely right. To see evidence for recurrence, though, it seems to me that if you see activation earlier in one site, and then a site further along the stream, you know, physically further forward, an activation there, and then activation here, where does this, so if activation has been originally here in this posterior site, it's died down while things are going on, and then it peaks again, what's happening to it? Where does that peaking come from? What's it due to? I mean, are you trying to evoke this explanation also for just visual effects? Because, I mean, I think there's quite a lot of evidence that in the visual system, you do have this recurrent activity. Or it's quite... Oh, yes. Yes.
You can save it for your question and answer session.
Okay.
So with that, let's thank Lolly for a fantastic talk. And we have coffee, posters. Please be back promptly at 4:15. And 3 lucky people, go run and get your tickets.